Hello guys, welcome back to my channel. In this video we'll be doing the respiration in plants chapter of standard 11 biology. We'll do it in two parts. In this video we'll do the first part, that is glycolysis and the fermentation. In the next video we'll do the remaining part, that is the Krebs cycle or the tricarboxylic acid cycle and the electron transport system. So before that, if you are new to my channel, then please subscribe it and click on the bell icon. Okay, so let's begin. Today we'll do the chapter 14 of 11th standard biology, and the chapter is respiration in plants. Okay, so let's start with this. This is the introduction part. Okay, so we'll directly start. There is nothing important in this. We'll start with okay, here from this part. All the energy required for life processes are obtained by oxidation of some macromolecules that we call as food. This you all know, right? Wait, just a minute. Okay. Only green plants and the cyanobacteria can prepare their own food because they have the chlorophyll, they have the pigments to trap the light, and by the process of photosynthesis, that is, they trap sunlight energy and convert it into chemical energy that is stored in the bonds of the carbohydrates like glucose, sucrose, starch. So they basically prepare glucose, which is stored in the form of starch. Okay, so we must remember that in green plants too, not all the cells, tissues and organs photosynthesize. Only the cells containing chloroplast, that is the mesophyll cells, guard cells, they have the chloroplast. Not all the cells, only few. And they carry out photosynthesis. Hence, even green plants, all other organs, tissues and cells that are non-green, they need food for oxidation. All that is, everything, all the living things, they require food. Because the food may be different in the different forms, but they all require it. Okay, so even those cells which do not photosynthesize, which lacks the chloroplasts and all pigments, they still require food for their oxidation. Hence, food has to be translocated to all non-green plants where the food is not synthesized. We need to transport that food. So, manlo, if you are sitting here in Goa and you want wheat, and suppose wheat is not cultivated here, then we need to import the wheat from other states or the other countries. So in this way, here in plants also they need to import it from other cells. Okay, similarly, if you have something in your, like in your state or in your country, then we export it to other countries. Similarly, those cells which prepared food, they export it to the other cells, they transport it, or we say it is translocated through xylem and phloem. And animals are heterotrophs, so they require food. They can't prepare their own food. They are not autotrophs, they are heterotrophs, and they obtain food from plants directly, are called herbivores. Those obtain food from plants directly are called as herbivores. They are directly dependent on the autotrophs like plants. Okay, like cows, all the grazing animals. Okay, cows, sheep, goats, etc. And there are some those are dependent on the other organisms, on the other animals, which are in turn dependent on these plants. They are called carnivores. Okay, like tiger feeds it is a predator. Okay, and the prey is, let's suppose, cow or a goat or a deer. So they are all herbivores. Saprophytes like fungi are dependent on dead and decaying material. We all know this. So what is important to recognize is that ultimately all the food that is required for life processes come from the photosynthesis. Finally, everything comes from the light energy. Energy gets converted into chemical energy, that is food, that is through photosynthesis. And then finally we get food from the other animals or directly from the plants. So the ultimate source is this photosynthesis. And this chapter deals with the cellular respiration or the mechanism of breakdown of food. What is respiration? It is breakdown of food materials within the cell to release the energy that is trapped in the form of ATP, the bonds in the ATP. Okay, so photosynthesis, of course, takes place within the chloroplast in the eukaryotes, where the breakdown of complex molecules to yield energy takes place in the cytoplasm. Okay, and in the mitochondria. So first step, that is glycolysis, which is common to all the organisms, it is found in the cytoplasm. It is the only mode of respiration in the anaerobes. And the next step, that is Krebs cycle or the tricarboxylic acid cycle, TCA cycle, it takes place in mitochondria, that too only in eukaryotes, and those who are aerobes. Okay, the breaking of, because mitochondria is organelles, and these membrane bound organelles are found only in eukaryotes, not in prokaryotes. Okay, that's why it is called only in the eukaryotes. The breaking of CC bonds of complex compounds through oxidation within the cells, leading to release of considerable amount of energy, is called as a respiration, that is breakdown process. The compounds that are oxidized during this process are known as respiratory substrates. What are they known as? They are known as respiratory substrates. 1 minute, students. So the compound oxidized during this process are known as respiratory substrates. Usually carbohydrates are oxidized. We all know this. And they release energy, but proteins and fats, even organic acids, can be used as a respiratory substances in some plants. But the most common is the carbohydrate. Okay. So there are different terms based on the respiratory substrate. If suppose carbohydrates and fats are used, it is called as a floating respiration. If proteins are used, it is called protoplasmic respiration. Okay, and these organic acids and all those proteins and fats are used only under certain conditions. It is not generally they use only carbohydrates. During oxidation within the cell, all the energy contained in a respiratory substrate is not released free into the cell or in a single step. Okay, and it is released in a series of slow stepwise reaction controlled by the enzymes, and it is trapped as a chemical energy in the form of ATP. This you all know, we have studied this in the lower classes. Hence it is important to understand that energy released by the oxidation in respiration is not, or rather cannot be used directly, but is used to synthesize ATP. Okay, the energy which you get after breaking the bond, which is released, it is stored in the form of ATP. You can't directly use it, and this ATP is broken down whenever the energy is needed to be utilized. Okay, and hence ATP acts as the energy currency of the cell. This you all know. And mitochondria is called as the powerhouse of the cell. The energy trapped in ATP is utilized in various energy requiring processes of the organisms, and the carbon skeleton produced during the respiration is used as a precursor for biosynthesis of other molecules in the cell. Okay, do plants breathe? We all know that animals breathe. We have done breathing here means the pulmonary respiration, that is we have we inhale the air, then we exhale the deoxygenated rich this carbon dioxide rich air. So this is breathing. So, well, the answer to this question is not quite so direct. Plants require O2 for respiration to occur, and they also give out CO2. Generally they take in CO2 for the photosynthesis, but they also give out CO2. Hence plants have systems in place that ensure the availability of O2, that is the oxygen. Plants, unlike animals, have no specialized organ for gaseous exchange. You know that we have lungs, we have all the trachea, bronchi, nose, all this respiratory system we have for the gaseous exchange, but plants don't have such uh, organ system. And they have stomata and lenticels for this purpose. Lenticels help in gaseous exchange as well as in transpiration. Even stomata are meant for the gaseous exchange. Okay, there are several reasons why plants can get along without a respiratory organ. First, this is very important. Each part of the plant, each plant part takes care of its own gaseous exchange needs. Okay, there is very little transport of gases from one plant part to the other. So first reason is why they don't require such organs for breathing is each plant part can take care of its own gaseous exchange needs. In our case it is different. We have only one respiratory organ. I mean the only one organ through which we take the air in. Okay, and each part of the body cannot take part in this gaseous exchange in animals, but in plants they can take part. And secondly, the very little transport of gases is required from one part of the plant to the other. It is because each part can take care of its own exchange needs, so there is no need of transport across long distances. But in our case, the air which we take in, it needs to be transported to the other parts. Like we each cell of our body require oxygen, okay, for living. Even the cell which is present on the tip of your toe, it requires oxygen. And you just see how long the distance it has to cover from the lungs to the tip of the toes, okay. So second reason is plants do not present great demand for gas exchange. This is the second reason. Roots, stems, leaves respirate rates far lower than the animals do. So their respiratory rate is very slow. They don't present great demand for respiration. I mean for gaseous exchange. Only during the photosynthesis large volumes of gases are exchanged. Only during photosynthesis they require CO2, they give out O2. This is the gaseous exchange. And each leaf is well adapted to take care of its own needs during these periods. So we have already discussed this, that each part can take care of its own gaseous exchange needs, and there is very little transport of gases in plants. The second reason is uh, they don't present great demand for respiration. Only during photosynthesis they require such large number of volumes of gases, and uh, the leaf is well adapted to take care of its own needs during these periods. When cells photosynthesize, availability of O2 is not a problem in these cells since O2 is released within the cells. Okay, so this is one more reason for this. And the third is the distance that gases must diffuse even in large bulky plant is not great because they have stomata, they have lenticels, almost like they don't have only one stomata or one lenticel, they have thousands and millions of such things. So they don't require the distance to which this gases diffusion take place is very small. Okay, distances that gases must diffuse even in the large bulky plant is small. Each living cell in plant is quite close to the surface of the plant, and this is true for leaves, you may ask. But what about thick woods, stems and roots? In stems, the living cells are organized in thin layers inside and beneath the bark. And they also have openings called lenticels on bark, right? And the cells in the interior are dead, so they don't require O2 and all. The innermost cells, they are all dead cells. The xylem, the xylary elements all are dead cells. Okay, the heartwood, it is a dead cell. And they provide only the mechanical support. Thus, most of the cells of the plants have at least part of their surface in contact with the air. One thing, because of they are very close to the surface of the bark and they have lenticels. This is the reason for that. This is also facilitated by loose packing of the parenchyma in the leaves. Okay, in dicot leaves generally the upper is palisade parenchyma and the lower one is the spongy parenchyma. The spongy parenchyma has the air spaces in it, so it is facilitated by this loose packing of the parenchyma in the leaves, stems and the roots, which provide the interconnected network of air spaces. The complete combustion of glucose, which produces CO2 and H2O as the end products, yield energy, most of which is given out as heat. If this energy is to be utilized, it is useful to the cell. It should be able to utilize it for the synthesis of other molecules that the cell requires. Strategies that a plant cell uses is to catabolize the glucose molecule in such a way that not all the liberated energy goes out as heat. All energy should not go out as a heat. They need to trap it or store it in the form of ATP. Okay. The key is to oxidize glucose not in one step but in several small steps, enabling some steps to be just large enough such that the energy release can be coupled to ATP synthesis. How this is done is essentially the story of respiration. The synthesis of ATP and all, this is all in the respiration part. During the process of respiration, oxygen is utilized and carbon dioxide, water and energy are released as the products. We all know that. Why do we need oxygen then? We need it for respiration, and we give out CO2, even water. Okay, and the energy is released. The combustion reaction requires oxygen, but some cells live where oxygen may or may not be available. The anaerobes. Okay, can you think of such situations and the organisms where O2 is not available? There are sufficient reasons to believe that the first cells of this planet, they lived in an atmosphere that lacked oxygen. It was reducing atmosphere at that time. We all know that, and there was no free oxygen in the air, so they were anaerobes. Even among present day living organisms, we all know of several that are adapted to anaerobic condition. Some of these organisms are facultative anaerobes, which means they are generally aerobes, but they can live even without oxygen, just like mycoplasma, the smallest living cell, which is a bacteria. It lacks cell wall and it is facultative anaerobe. It is born as aerobe, but it can live without oxygen. And in sum, the requirement for anaerobic condition is obligate. They are called obligate anaerobe. They cannot survive in the presence of oxygen. They require completely anaerobic condition. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. So the initial step doesn't require oxygen. It is partial breakdown of glucose to pyruvic acid. This is called glycolysis, and it is present in all the living organisms. It takes place in cytoplasm, not in any cell organelles, and it is the only mode or the process of respiration in the anaerobes. This breakdown, the partial breakdown of glucose to pyruvic acid, is called glycolysis. This is the first step of the respiration. So now let us start with the glycolysis. The term glycolysis has originated from the Greek word glykos, which means sugar. Glykos means sugar. Okay, it is a Greek word. Remember this. It might come for the need. And the lysis is for splitting. The scheme of glycolysis was given by Gustav Emden, Otto Meyerhoff, and J. Perenas, and is often referred to as EMP pathway. It is called EMP pathway. In anaerobic organism, it is the only process of respiration because they lack they don't require oxygen. And glycolysis also doesn't require oxygen. It takes place in cytoplasm. It does not require organelles also. So it is the only mode of respiration in anaerobes. Glycolysis occurs in the cytoplasm, and it is present in all the living organisms. It is the process in which glucose undergoes partial oxidation to form two molecules of pyruvic acid. Glucose is a 6 carbon molecule compound and pyruvic acid is 3 carbon molecule compound. So there is formation of two molecules of pyruvic acid from one glucose molecule, which is the end product of the photosynthesis, derived from the sucrose. Okay, wait. In plants, this glucose is derived from sucrose, and sucrose is the end product of photosynthesis, or from the storage carbohydrates like starch. Sucrose is converted into glucose and fructose by the enzyme invertase, also called as sucrase, and these two monosaccharides readily enters the glycolytic pathway. We all know glucose and fructose are isomers of each other. They are interconvertible. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of enzyme hexokinase. The phosphorylated form of glucose is then isomerized to produce fructose 6-phosphate. Okay. Subsequent steps of the metabolism of glucose and fructose are same. The various steps of glycolysis are depicted in figure 14.1. In glycolysis, a chain of 10 reactions. It is not a single step. It is chain of 10 reactions under the control of different enzymes taking place to produce the pyruvate from the glucose. What is the final product? That is the pyruvic acid. We want pyruvate, the pyruvic acid. So in this, you can easily see here it's given glucose, which we get from the sucrose. Breakdown of sucrose by sucrase gives glucose and fructose. So glucose is converted into glucose 6-phosphate by hexokinase, and here ATP is used because there is addition of phosphate. Then glucose 6-phosphate to fructose 6-phosphate, they isomerize. Then fructose 6-phosphate to fructose 1,6 bisphosphate, that is 2 molecules of phosphate are there. So one more ATP is utilized here, and this is by the enzyme phosphofructokinase. Here the first step was by hexokinase, third step by phosphofructokinase. Then fructose 1,6 bisphosphate is converted into triose phosphate, that is glyceraldehyde 3-phosphate, and the other is dihydroxyacetone phosphate. So these two separate molecules are formed. They are again isomers, interconvertible. So let's take only two molecules of glyceraldehyde 3-phosphate instead of one molecule of this and the other molecule of the other one. We'll take only two molecules of this, any one of them. So here in the NCERT, glyceraldehyde 3-phosphate is given. So we'll take two molecules of glyceraldehyde 3-phosphate. It is converted into 1,3 bis-phosphoglyceric acid, and the NADH is synthesized here. So since there are two molecules, there will be synthesis of two molecules of NADH. Then 1,3 bis-phosphoglyceric acid is converted into 3-phosphoglyceric acid. So here one phosphate is reduced. That means ATP is synthesized from one molecule. Since there are two molecules, two ATPs. Then 3-phosphoglyceric acid to 2-phosphoglyceric acid, nothing. 2-phosphoglyceric acid to phosphoenol pyruvate, PEP. Here again, this is the only step of dehydration. This is the dehydration step. Here the water is given out, and this is by enzyme enolase. Okay, 2-phosphoglycerate is converted into phosphoenol pyruvate by the enzyme enolase dehydration process. Then phosphoenol pyruvate is converted into pyruvic acid. Again ATP is synthesized. So there are two molecules, as there are two molecules of PEP and two molecules of pyruvic acid. Now, while studying the steps of glycolysis, please note that steps at which the utilization or synthesis of ATP, or in this case NADH H+, it take place. ATP is utilized at two steps. We all know this. The first two steps, one is conversion of glucose to glucose 6-phosphate, and then fructose 6-phosphate to fructose 1,6 bisphosphate. It's given here. Okay. Now the fructose 1,6 bisphosphate is split into dihydroxyacetone phosphate and the 3-phosphoglyceraldehyde. We find that there is one step where NADH H+ is formed from the NAD+. This is when the 3-phosphoglyceraldehyde or 3-phosphoglyceric acid is converted into 1,3 bisphosphoglyceric acid. Okay. So this is 3-phosphoglyceraldehyde or glyceraldehyde 3-phosphate. It forms two molecules of 1,3 bisphosphoglycerate, and there is synthesis of NADH. Two redox equivalents are removed in the form of the two hydrogen atoms from PgAl, that is phosphoglyceraldehyde 3-phosphoglyceraldehyde, and transferred to a molecule of NAD+. 3-phosphoglyceraldehyde is oxidized and with inorganic phosphate to get converted into 1,3 bisphosphoglyceraldehyde. This conversion of bisphosphoglyceraldehyde to 3-phosphoglyceric acid. Okay. So this conversion is actually there is synthesis of ATP is also an energy yielding process from bis, that is two molecules of phosphate, 1,3 bisphosphoglyceric acid. It forms 3-phosphoglyceric acid. One molecule of phosphate is removed. So there is synthesis of ATP. Another ATP is synthesized during the conversion of PEP to pyruvic acid. So there are total 4 ATPs synthesized and 2 ATPs used in the first two steps. So there is net gain of only 2 ATPs in glycolysis, and there is net gain of 2 NADH+. Pyruvic acid is then the key product of the glycolysis. What is the molecular fate, metabolic fate of pyruvate? This depends on the cellular needs, that is whether the cell is aerobic or whether it is anaerobic. Here the pyruvate is now transported into mitochondria if it is the aerobic respiration. If it is anaerobic, then this is the last step, that is glycolysis only takes place in them, and then there is fermentation of pyruvic acid either to lactic acid or the ethanol in the alcohol fermentation. So there are three major ways in which different cells handle pyruvic acid produced by the glycolysis. These are lactic acid fermentation, alcohol fermentation, and the aerobic respiration. In both alcohol fermentation and aerobic respiration, CO2 is given out, but in lactic acid fermentation there is no CO2, only lactic acid is given. Fermentation takes place under the anaerobic condition in many prokaryotes as well as unicellular eukaryotes. Okay, for the complex oxidation of glucose to the CO2 NH2O. However, the organisms adopt Krebs cycle or the TCA cycle. This is called aerobic respiration, and this requires O2 in the terminal step, not in the initial steps. Okay. So here it is also given that apart from many prokaryotes, unicellular eukaryotes are also involved in fermentation. For example, yeast. Yeast is involved in the alcohol fermentation. It produces ethanol. And who is yeast? Yeast is a unicellular, and that is eukaryote. So guys, now let's look in this fermentation part. In fermentation, say yeast, that is a unicellular eukaryote, the incomplete oxidation of glucose is achieved under the anaerobic condition by sets of reaction where pyruvic acid is converted into CO2 and ethanol. That is alcohol fermentation. Okay, and the enzyme pyruvic acid decarboxylase and alcohol dehydrogenase are involved in alcohol fermentation. What are the enzymes involved? Pyruvic decarboxylase and alcohol dehydrogenase. They catalyze this reaction, and the other organisms like bacteria produce lactic acid from the pyruvic acid. The steps involved are shown in the animal cells also, like muscle cells. During exercise, when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid. This is called lactic acid fermentation by lactate dehydrogenase. In case of ethanol fermentation or the alcohol fermentation, enzymes involved are pyruvate decarboxylase and alcohol dehydrogenase. In lactic acid fermentation, it is lactate dehydrogenase. The reducing agent NADH H+, which is reoxidized to NAD+ in both the processes. So again, since there are two molecules of pyruvic acid, it will produce two molecules of lactic acid or the two molecules of ethanol, and so two molecules of NADH H+ are also used. So in glycolysis, two molecules of NADH H+ were synthesized, and again they are now used in the fermentation. So there is no net gain of NADH H+ in fermentation. So there is only net gain of 2 ATPs in fermentation. They were synthesized during the glycolysis process. In both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of the energy in glucose is released, and not all is trapped as the high energy bonds of ATP. Also, the processes are hazardous. Either the acid or the alcohol is produced. And what is the net ATP that is synthesized? Okay. So when one molecule of glucose is fermented to alcohol or lactic acid, there is net gain of 2 ATPs. And there is no net gain of NADH H+ because it is again used in the fermentation part. Yeast poison themselves to death when the concentration of alcohol reaches about 13%. This is the important line, and that's why the concentration of wine or a beer is 13%, but they can be further increased by distillation. Yeast poison themselves to death when the concentration is 13%. 13% is the maximum concentration. When then what then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think alcohol beverages of alcohol content greater than this concentration are obtained? This is by distillation. What then is the process by which the organism can carry out complete oxidation of glucose and extract the energy stored in the glucose and synthesize a large number of ATP molecules? We know that here only in fermentation only 2 ATPs are synthesized, and less than 7% stored in the energy in the glucose is released, and not all the energy is stored as a ATP. But in case of complete oxidation, that is aerobic respiration, large number of ATPs are synthesized, and they are needed for cellular metabolism. In eukaryotes, these steps take place within the mitochondria and requires O2. The aerobic respiration is a process that leads to complete oxidation of organic molecules or substances in the presence of oxygen and releases CO2, water, and large amount of energy present in the substrate. Around 38 ATPs are synthesized, and this type of respiration is most common in higher organisms. We will look at this process in the next session. Okay, so we'll also look at this in the next video. Till then, whatever is taught, whatever is done in this part, just read it and solve as many questions as you can on this part. This is a very important part for the NEET. Okay, so thank you for watching my video, and please like it, Subscribe my channel if you have not subscribed it, and share it. Thank you.